welcome Dr. Bedlock and welcome everybody. You know, today's event, I'm really, really excited to host Dr. Bedlock. And we have 300 sessions here and 300, I would say about 400 of you are here today to interact with Dr. Bedlock. And I don't think anybody needs another introduction of Dr. Bedlock because he's very unique. And I have to say, I actually looked for a lot of outfits today to see how can I wear something colorful, because I was wondering if I can beat Dr. Bedlock in his colors, but I don't think I did. So, you know, as colorful as Dr. Bedlock dresses, he brings those colors to our ALS community. And I would say we're very fortunate and also very, honored to have Dr. Bedlock and his unique perspective and his colorful thinking in our ALS community. And already, as McFinn said, might be 48 is a small number, but having those people reversed gives us hope that this disease can be cured, can be reversed, can be treated. So we're really excited. And Finn, somebody asked on the text that, you know, could we actually discuss McFinn's condition before he reversed? And that will be one of the things we want to bring to you, because McFinn will be moderating the session with Dr. Bedlock. And also joining us also with Sarah, who is our patient advocate, who had a personal experience with her father being affected. And she's also a medical student at Washington University. Who will be moderating the session, all the chats that the questions you've already sent, and the chat in the chat box. So feel free to ask questions on the chat box. If you can't get to it, we'll get to it later, or some of the other people can answer for you. So there's a lot of dialogue. Like I said, you know, we have close to 400 people here. So with that, I would actually welcome Dr. Bedlock. Please give him a warm welcome. And Dr. Bedlock, please, it's your floor right now.
Thank you so much, Indu. That's a very kind introduction. It's my pleasure to be with you tonight to tell you about this interesting journey that I've been on with these cases of what I call ALS reversals. And so what I thought I would do as far as organization is tell you a little bit about my experience with ALS progression before the year 2011. 2011 is when I first encountered what I now call an ALS reversal. And I'll tell you about that case. I'll tell you what I decided to do about cases like that in terms of how I'm defining ALS reversals, how I'm collecting them, why I think they're worth studying, some of the different hypotheses that I have for explaining these cases. And then I will spend quite a bit of time on the two research programs that I've organized at Duke, the ROAR program, which is called Replication of ALS Reversals, and the STAR program, the study of ALS reversals. These are programs I'm trying to use to understand these cases better and hopefully make things like this happen more often in our community. So I really knew nothing about ALS until about 1997. I remember I was a neurology resident at Duke and I saw my first patient. And I thought it was the most amazing story and the most incredible physical exam I had ever seen in my career as a doctor. And I remember going out to present the case to my attending. When you're a resident, you work under a more established physician. And that person came in and basically said, look, we don't know why this happens. Unfortunately, people with this disease always experience rapidly progressive disability and shortened survival. And there's really nothing we can do about it. You just have to go home and get your affairs in order. And then they just walked out. And I remember just sitting there stunned and looking at the patient and family and thinking, if I feel this bad, I can only imagine how they feel. And I remember driving home that day and thinking, I'll stay at Duke and I'll try to build a program for people to have options for living a longer life, a better life, and I'll build some research into that program to give them some hope. But regarding that comment about progression, in the first few years of my career leading the Duke ALS Clinic, I learned about the ways that we measure disease progression in ALS. Basically, it comes down to measures of function and measures of survival. And so the main measure of function is called the ALS Functional Rating Score. We're using a revised version of this score now. And so you'll see the abbreviation ALS-FRS-R. This is a questionnaire. It's got 12 items. Each item is a question about a specific motor function. And you can hopefully see the 12 questions there. But they include questions about speech, handling saliva, swallowing, handwriting, cutting up food, dressing, walking, climbing stairs, and different aspects of breathing. And each one of these questions gets scored between 0, which means there's just no function there at all, up to 4, which means that that function is normal. So a completely normal score for a patient would be 48. And somebody who had no function at all would be a 0. I don't think I've ever seen anyone with a 0 in 20 years. If you look at large populations of people with ALS, the decline is pretty linear on average in this scale. So across a large population, the average decline on this scale would be about 1 point per month. And then there's that other measure that I told you about, which is survival. And it turns out to be a little controversial how to measure survival in ALS. You would think of all the things in the world to be controversial, deciding how long someone lived would not be that hard. But it turns out to be, because we do a lot of things that keep people alive now somewhat artificially. And so they don't always die from the disease. So for example, some people use non-invasive ventilation 24 hours a day, 7 days a week. And without that, they would have died much sooner. Some people get a tracheostomy and get attached to an invasive ventilator. And without that, they would have died much sooner. So measuring survival is somewhat different depending on who you talk to. But I personally think the best definition in our field is tracheostomy-free survival. How long can you live without being attached to a ventilator? And on average, that's about 3 years from symptom onset. But it became very clear to me, even after just a few years of taking care of people, that there's a lot of variability between patients with this disease. So we can talk about variability in functional measures. On the left-hand side of this slide, you can see about 40 patients, their ALS-FRS-R scores over time. And you can see that some of those lines are almost straight down. They lost function very quickly. And some of those lines are very flat. Function took a long time to get lost. There's also a lot of variability in tracheostomy-free survival. So I said that on average, people live 3 years, but 20% of people live more than 5 years. 10% live more than 10 years. 5% live more than 20 years. And there are, pretty famous examples of people living 50 years or more with this disease, like Stephen Hawking. So it took me a few years to realize that, but I think, most people now, most doctors and most patients understand that there's differences between patients. But one thing that took me even longer, and one thing that I think is still not widely understood by doctors or patients, is that there can be variability in the course of the disease within one patient at different times. And so I had a theory about this from taking care of so many patients, and I got together with some of my colleagues from Mass General Hospital and from the Patients Like Me group, and we did a big study of all the people that were enrolled in something called the PROACT database. So in this database, there's hundreds of patients with ALS that have participated in previous clinical trials, and there's all kinds of things measured over time. So the most common measurement, of course, is the ALS-FRS-R score. And so I can show you what this looks like on an individual patient's ALS-FRS-R score over time. That's what's in the upper right-hand corner of this slide. This is one patient in the database who had been randomized to a placebo. This is what their score did over 500 days. And I think it's pretty easy to see that is not a straight line down. There's periods when that patient progressed a lot, and there's periods like between day 400 and day 500 where they didn't progress at all. So we can look at the whole database instead of looking at one individual patient, and that's what I'm trying to show down here in the lower right-hand corner. And so these different curves are different degrees of ALS FRSR stability or improvement lasting different amounts of time. So let me take you through this. This blue line is a stable ALS FRSR score. And what you see here is the percentage of people in the database whose score was stable over different amounts of time. So there's a lot of people who had a stable score over just 1 month. There's very few people who had a stable score over an entire year. The green line is a 1-point improvement in that scale versus how long it lasted. The red is a 2-point improvement. The aqua blue is a 3, and the purple is a 4-point improvement. And again, for each of those degrees of stability or improvement, it's much more common for that to happen over very short periods of time. So that was new to me, but it turns out that it had been demonstrated many times before. In fact, these types of periods of plateaus or small improvements were even demonstrated in the career of Lou Gehrig. There's a new book that came out last year called Last Ride of the Iron Horse, which I thought was terrific. And it really analyzed the last full year of Lou Gehrig's playing career, which was 1938. And one of the amazing things is that Lou Gehrig appeared to have a month where he all of a sudden got significantly better in the midst of his ALS. And so let me try to prove that to you. So there's 2 ways that we look at the career of a baseball player like Lou Gehrig in terms of their offensive performance. One is the batting average. So that's the total number of hits divided by the number of at-bats. So if you're 333, that means you get a hit 1 out of every 3 times you come to the plate. And then there's something called the slugging percentage, which is a measure of power. And that's the total number of bases divided by the number of at-bats. So if you only had 1 at-bat and you got a home run, that number would be 4.0. And so take a look at Lou Gehrig's career numbers here. Batting average for his whole career was 340. Slugging percentage was 632. In 1938, for most of the year, he was way below those numbers. His ALS, we think, started around April of that year. And between April 18th and August 6th, his batting average was only 274, 70 points below his career average. And his slugging percentage was down 200 points. But then mysteriously in the month of August, he exploded. His batting average went up to over 350. Slugging percentage went up to over 700. You know, those are much better than his average for his whole career. Unfortunately, that improvement didn't last. So, you know, by later in the baseball season into the World Series, he was back down to being well below his average. So it's really important for folks to understand that people with ALS do not always progress on a straight line, that there can be periods where they don't progress at all or where they even improve a little bit for a while. It makes it really difficult to analyze the effects of any new treatment based upon what I just told you. But one thing I never expected to find, and I didn't believe could exist until 2011, is something that I call an ALS reversal. And so here's the first case that I ran across. This is a woman named Nelda Boos. She's made her name public, so there's no privacy issues here. I came across her in the midst of an ALS Untangled review. Most of you probably know what ALS Untangled is, but it's kind of like my own version of the X-Files. You know, people submit ideas to me about things they've read on the internet that might help ALS, and I send my investigative team to look at these things, and we try to really look at them critically like scientists do, and we try to write reports of what we find. And the whole point of it is to help patients make more informed decisions of some of these unusual ideas that are out there. And so I came across this lady in the midst of an investigation on energy healing, specifically on the website of a man named Dean Kraft, the energy healer. There was a video of this woman saying that she had been diagnosed with ALS at age 43 and that she progressed rapidly and that within 6 months, she really couldn't walk or take care of herself. And within a year, she was totally paralyzed. She couldn't move either arm or either leg, and she was short of breath. And at that point, she said she was told by her ALS clinic there was nothing more to do it was time for hospice. And so she mentions in the video that she decided to take one last chance and go see this energy healer. And over the next 2 years, she documents her improvement back to completely normal. And she's remained completely normal, working on a farm, climbing ladders, stacking firewood for more than 30 years since this happened. So I thought the video was pretty amazing. And as you all know, you can't believe everything you see on the internet. So the first thing I did is try to find Nell DeBoos. And I found someone who knew her, put me in touch with her. She was kind enough to send me a big box of her medical records. And I'll never forget the feeling I had when I was about halfway through. I turned to my wife and I said, I don't believe it. I mean, this lady really had ALS. Clearly, incredibly well-documented history, well-documented neurological exams, showing progressive weakness, showing widespread upper and lower motor neuron signs, clearly documented EMG studies showing widespread loss of motor neurons, and lots of tests to try to find other explanations for what was happening that showed no abnormalities. And all these things were done by people that I personally know at the University of Virginia, doctors that I have a great deal of respect for. So it was very clear to me that she had ALS and that she really did progress and then really did improve the way she said in the video. And so I had a decision to make. What do I do with this case? Do I actually, submit it for further study? And of course, you know the answer, but back then my thought process was, we kind of need some new models of ALS. And I still believe this. The models that we have, I don't think they're great. So, we have all these animal models that are made by inserting genetic abnormalities into mice, rats, flies, fish, worms. And, those animals do show signs that are similar to a person with ALS. They do get progressively weak and they do have shortened survival. But the cause of it is not a cause that most people have. Most people don't have a genetic abnormality causing their disease. And, we've screened many drugs in these animals. We found many things that dramatically slow progression and prolong survival in these animal models. And so far, none of those things have worked in people. And, in the last few years, there are a lot of other models that people are coming up with that I think might be better. Like one model I'm excited about is the model of induced pluripotential stem cells, where they actually scrape the skin of a person with ALS and they turn those skin cells into stem cells. And then with chemicals, they can turn those stem cells into any kind of cell they want, motor neurons, astrocytes, microglia. And so for the first time, we can actually study cells from the central nervous system of people with sporadic ALS. And I think that may turn out to be a better model, but only time will tell. Clearly, we need to have as many different models as we can for studying this disease. I also think that this model is relevant because something similar happened in another field and it led to a medical breakthrough. So back in the 1980s, it was recognized that some people were infected with the HIV virus, but they never got sick. And even when, really good antiviral medications came along, these people didn't take them. And they still seemed to be able to control the disease. It wasn't making them sick. They got nicknamed the elite controllers. It's about 1% of everybody who gets infected with the virus. And for a long time, people just kind of shrugged their shoulders and said, I don't know what's going on with them. And then finally somebody said, well, we probably should study it. And they put them all in the same database. And one of the things they did was genetic testing. And it turns out that many of these elite controllers have the same genetic abnormality. It's in a gene called CCR5. They have a slight variation in that gene, which is called the delta 32 variation. And so scientists said, well, that's interesting. What does that gene do that might have anything to do with ALS? And it turns out they learned that that gene codes for a protein that's on the surface of all cells, which you can see in this picture right here, CCR5 protein. HIV needs that protein to get into cells and make people sick. And as a result of the mutation in these patients, that protein was bent and HIV couldn't get into the cells. And so a drug company, Pfizer, seized upon that and developed a drug that would act on that receptor and prevent HIV from getting into cells. And it actually works. It helps everybody to be more resistant to HIV. So why not study these ALS reversals and see if it could lead to a breakthrough like this? Maybe there's something they have in common that we could exploit to create a treatment that would reverse ALS in everyone. And so the first thing you have to do when you want to study something is figure out how to define it. And so I decided to set a very high bar for calling something an ALS reversal. And there's 3 parts to it. All these involve me getting the medical records and reviewing them. The first is I have to agree with the ALS diagnosis. So the records have to show me the right history, the right neurological exams, the right pattern on the EMG, and the right set of tests to rule out things that can mimic ALS. Second, the records have to show me objective evidence that the disease progressed to where the person was disabled from it. And third, there has to be evidence of a dramatic and a persistent recovery of lost motor function and at least some disability. So some examples of things that I've called ALS reversals. I found patients who were dependent, they had a tracheostomy and they were attached to a ventilator to stay alive. And they got better enough to be able to be weaned off the ventilator and now breathe on their own. I've got examples of patients who couldn't swallow and had to take all their hydration and nutrition through a feeding tube, and now they swallow normally. I've got examples of patients who lost their ability to speak and had to use a speech generating device, and now they talk normally. I've got examples of people who were wheelchair bound, and now they can walk and even run. And I've got examples of people who were completely dependent upon others for their activities of daily living, for dressing and feeding, and now they do those things on their own. So those are the kind of things that I'm calling ALS reversals. And so how do I find these? Well, I'm advertising. I've got a website up, alsreversals.org. I'm doing talks like this. It's part of the reason I love to do these talks, is because usually after one of these talks, I get a few more sets of records to look at. I'm even trying to give webinars like this to other doctors. And as a result, I think of this publicity of this program, I've been able to collaborate with people around the world to confirm 48 of these cases of ALS reversal. And so there's a lot of different possible explanations for these. The first one, the one that most other doctors think is the right one, is that these people never had ALS in the first place, that they were misdiagnosed, that they had something else that looked so much like ALS that even an expert can't tell it apart. If that's the case, I don't know what it was. It's something that's never been described before. And that's really hard to test for. The second possible explanation is maybe there's something different about these people, like the HIV elite controllers, something that made them resistant to this disease so that they got a form of ALS that their body could fight off and recover from. And the third possible explanation is that maybe some of the things these people did to themselves, some of the treatments they started actually worked. And so let's start with the mimic idea first. I personally do not think these were mimics. And the reason is, ALS was first described in the 1800s. You would think if there was a mimicker of ALS, it's been around as long, somebody would have figured it out and described it by now. These patients, I've looked at their records. They've got classic stories, classic exams, classic EMGs, lots of testing for known mimickers, and it's all consistent. 4 of the 48 people that I found in this database have a family history of ALS. That's about the same percentage that we see in large databases of people with more typical ALS. So if all these people have a mimic, they have something that's not ALS, why would 10% of them have a family history of ALS? It just seems like too much of a coincidence. But finally, and maybe most convincingly, the way I imagine these people recovering is exactly the same way that I would imagine a person recovering from polio. And let me explain that to you. It's through a process called collateral sprouting. And so this is sort of a really simplified version of what happens. So let's imagine that this thing that looks like railroad tracks is a muscle. And in a healthy person, that muscle is innervated by 4 motor neurons. And now you get sick. So for example, you get polio, and it wipes out some of these. So in this example, it wipes out 1 of the motor neurons that go to that muscle. And as that motor neuron dies, part of that muscle does not have any connection to the nervous system. And so the muscle is going to be weak. But in the case of polio, if a person survives the virus, they recover. How do they recover? They don't recover by making a new motor neuron. You can't make new motor neurons. They recover by the motor neurons that were next to the dead one sending off branches, so that eventually the whole muscle is reconnected to the nervous system, except it's reconnected by a smaller number of motor neurons. Now, how do I know that my ALS reversals recovered like this? Because many of them had EMGs done after they recovered. And their EMGs looked exactly like the EMG of a person who recovered from polio. They had a much smaller number of motor neurons driving their muscles. And so it wasn't like some magic thing happened. Exactly what happens in polio happened to these folks. And so I mentioned in the beginning that I've got 2 different broad programs underway to try to work through some of these hypotheses. The first one is called STAR, Study of ALS Reversals. And there's different parts to this. Again, we're going to try to find as many more of these cases as we can. We're putting them all for the first time into a database so that we can compare their demographics, their disease characteristics, the types of medical problems they had, the medications and supplements they took, to people with more typical ALS who are in other large databases, like, for example, PROACT that I showed you before, and also the National ALS Registry. We're trying to get biological samples on as many of these people as we can. And we're going to do a lot of different things with them: genetics, microbiome. I'll tell you more about that in a minute. And we're trying to ask as many different questions to these people as we can to understand, what might have been unique about their environment before and during their ALS reversal that might have contributed to it. And so I can tell you that we know a few things. Right now, we know that ALS reversals are younger than the average person with ALS. So in large databases, the average person with ALS is about 60. The reversals are about 50, with a range of 18 to 75. Just like large databases of people with ALS, ALS reversals are more likely to be male. So 39 out of 48 ALS reversals were men. Just like in large databases, ALS reversals are more likely to have limb onset versus bulbar onset disease. 44 out of 48 had limb onset. Interestingly, the people with ALS reversals tended to progress faster than the average person with ALS before they started to improve. I don't know what that means yet. And compared to patients with more typical ALS in large databases, it looks like ALS reversals are more likely to have taken certain types of treatments. And so this is from an analysis that we did of the first 39 or so reversals. And you can see some of the treatments that are much more common in these reversals. And at the very top of the list, one we're going to talk about some more is curcumin. And so there were far more ALS reversals taking curcumin than we could find in databases of more typical ALS. We want to look at the genetics of these folks, as I mentioned before. And so we're doing that by collecting saliva samples. And we have saliva now from most of the ALS reversals. And that's been sent to some collaborators we're working with at the University of Miami. They're part of the National Institute of Health's CREATE Consortium. And what they're going to do for us is something called whole genome sequencing. So they're going to look at every single gene that can be looked at in these ALS reversals and compare all the genes to people that they have on file with more typically progressive ALS. And, as I said before, if I had to pick one theory that I think I'm most excited about, it's this one. And that's just because of what happened in those HIV elite controllers. If we can zero in on a gene that's different in these ALS reversals that confers this ability to get rid of ALS, nowadays, once we know about a gene, we can quickly figure out what it does and design a drug that does that in everybody. So this might be a way to quickly latch onto what happened in these people and make it happen more often. Oh, by the way, we should have the results back sometime in 2020. We got a little delayed with the pandemic, but we're hoping to have those results back by the end of this year. So the second STAR study is environmental exposures, and this is a collaboration with the National ALS Registry. And so those of you that have participated in the National ALS Registry know that one of the things they're doing is something called a risk factor survey. They're asking lots of questions about the lives of people with ALS, things you might have been exposed to, jobs that you might have had. And so we decided to give this exact same questionnaire to our ALS reversals and compare the answers. And so what you see on this slide is the results from the first 19 ALS reversals. And out of all the questions, there was only one question that came out different in the ALS reversals. And that was related to the longest job held. ALS reversals were much more likely to have been woodworkers of some kind, carpenters or cabinet makers, compared to people with more typically progressive ALS. And we don't know what that means yet, but we have some theories, which I'll talk about later. The next STAR study is neuroimaging. And this is a collaboration with Duke neuroradiologists. So most of the ALS reversals on their way to getting diagnosed with ALS in the first place had lots of pictures taken of their brains and spinal cords. And we're going back over all those pictures with a fine-tooth comb. And we're trying to figure out, how many of these ALS reversals had some of the classic findings that we see in people with ALS. And so what do I mean by that? Well, here's an MRI scan of a person with sort of typically progressive ALS. And we don't see this all the time for some reason, but if we're going to see anything on the MRI, what we tend to see is abnormalities in a group of cells called the cortical spinal tract. Those are the upper motor neurons. And so in this picture, at the end of the arrows, you can see this sort of white area. Those are the cortical spinal tracts in this patient, and they shouldn't be white. They should just be a gray color like everything else. So something's wrong with those cortical spinal tracts in this patient. So we're going to look at these images very carefully and see, do we see the classic changes of ALS, or is there some other important clue on these pictures that tells us why these people got better? We've got a collaboration now with the National Institute of Health's Brain Bank. And this is allowing ALS reversals to donate their bodies when they die. Hopefully this won't happen for many years, but if these ALS reversals someday die from hopefully old age, natural causes, we want to look carefully at their brains and spinal cords. There's certain things that you see in the brain and spinal cord of people with ALS that are fairly unique. And here's some pictures of some of those things. So on the left-hand side of these pictures, these are all sections from the brain and spinal cord of people who died from ALS. And on the right-hand side, these are sections of normal, healthy people who died from natural causes. And you can see that inside the motor neurons, there's these funny looking blobs. These have different names, but these are clumped proteins. These are inclusions that shouldn't be there. And we want to know, do these ALS reversals have these kinds of changes, or is there some other important clue in the histology that tells us why they got better? We've got an upcoming collaboration that I'm very excited about, and that's with the Duke Microbiome Center. And this is to study the microbiome in people with ALS reversals. So for those of you that haven't heard of this, it's really amazing. The microbiome is the family of bacteria and viruses and funguses that live in all of our GI tracts, in our stomach, small and large intestine. And it's been known for years that this family of organisms can contribute to diseases of the gut, like ulcerative colitis and Crohn's disease. But amazingly, in the last few years, there's starting to be evidence that this might actually play a role in neurodegenerative diseases, like Parkinson's disease and even ALS. And so why do we think that? Well, first of all, because there actually are bacteria that we found that can secrete things that are neuroprotective. For example, something called butyrate. And there are other bacteria in the microbiome that can secrete things that are destructive to neurons. things that ramp up neuroinflammation, for example. There's even bacteria in our own guts that can secrete a toxin called beta-methylaminoalanine, BMAA, which is believed to be the cause of some cases of sporadic ALS. There have been 3 small studies of people with ALS looking at the microbiome, comparing it to healthy controls. And those 3 studies all showed differences. The problem is that the differences were different. At this time, I think it's fair to say that the microbiome may be different, but we don't know exactly how it's different. But the most amazing thing in the past couple of years is that now 3 different groups have looked within some of those different animal models of ALS. And all they've done to those models is manipulated the microbiome. And if they put in certain bacteria, ones, for example, that secrete healthy chemicals like butyrate, they can dramatically slow the progression of the disease in those models. And if they put in other bacteria that can ramp up inflammation or secrete toxins that are damaging to the brain, they can dramatically speed up progression. So to me, it's becoming very clear that the microbiome may play a role in ALS progression. And I would be fascinated to see how it might look different in ALS reversals compared to people with more typical ALS. We've got other ideas that we're trying to find collaborators for and funding for. So we would love to be able to send someone out to all the ALS reversals around the world and get blood samples. And that would allow us to look at RNA, protein, and measures of the immune system like cytokines to see what might be different compared to people with more typically progressive ALS. If we could get blood or skin, we could make those induced pluripotential stem cells from these ALS reversals. And that would allow us to really examine the behavior of their motor neurons and their glial cells in a dish, which I think would be fascinating. People have suggested that we look at the diets of these ALS reversals. And so we're trying to figure out how we might do that. It's going to be surprisingly challenging for 2 reasons. One is that there's no universally agreed upon way to measure diet. And so we're going to have to come up with something that captures all the different unusual diets that people with ALS sometimes try. The other problem is that in some of these patients, the ALS reversals occurred 20 or 30 years ago. So we'd be asking people to remember what their diet was 20 or 30 years ago, which may be difficult. And then finally, people have suggested that we might try to study the attitudes of ALS reversals. I've heard it suggested that maybe someone's attitude might have a role in how well they do with this disease. And it's possible, but it's a difficult thing to study. And, you know, whenever I'm trying to think of how to study something, I'm also trying to think, you know, how would I change that? So if I found that there was a difference in attitude, how would I give people a good attitude? I mean, there's a lot to think about there, but it's something that we're working on. So the other sort of broad program that I'm doing to study these ALS reversals revolves around this theory. Maybe they got better because of the treatments that they decided to take. And, you know, for some that might sound far-fetched, but believe it or not, there's many examples throughout history of patients discovering drug effects that researchers had never thought of. There's a whole paper about these, which I've included on this slide. It's called The Role of Serendipity in Drug Discovery. The most famous example is Viagra. So Pfizer was studying Viagra to see if it could help people who had heart disease. They didn't see anything happening and they were about to throw the drug in the garbage when the men who participated in the study came forward and said, I'd really like to keep taking this drug, and here's why. And the researchers kind of went, it does what? They had never thought about it being used for erectile dysfunction. And so they completely changed gears, put it in a trial for that. It worked. It became the first in-class drug for that problem. It was a $1 billion blockbuster drug that was discovered by patients. Nuedexta is another example. Nuedexta was being studied to see if it could slow disease progression, and it didn't. And we were about to give up on it, but patients in the study came forward and said, wow, you know, I used to have a problem controlling my laughing and crying. You know, you called it pseudobulbar affect, and it's gone. Nuedexta seems to have taken care of it. We had no idea it would do that, but we went back to the drawing board and did studies for that problem, and it works. And so now it's FDA approved for that problem. Again, we never thought of it. Patients discovered it. And so I've created this ROAR program, Replication of ALS Reversals, to do small pilot trials of some of the treatments that are associated with these dramatic ALS reversals. And, you know, because most of these things are food supplements, they're regarded by the FDA as safe. And because we're looking for such a large signal, we're not looking for a 10% slowing in progression. We're looking to see a reversal. We're looking to see people have a dramatic recovery of lost function. We can do some very different things with these trials that I think patients will really like. First of all, these can be very easy to get into. You know, why do trials have, you know, such difficult rules? Why do they have such narrow inclusion criteria? Because they're trying to homogenize a very noisy disease in order to look for a small signal. As I said, we're not looking for a small signal, so we can take just about anybody. We don't need a placebo. Why do you need a placebo? Again, you need a placebo if you're looking for small effects. If you're looking for effects so large that people get out of their wheelchairs and walk again, you don't need a placebo. People would never say that was a placebo effect. We can use historical controls in these trials. We probably don't need in-person visits. We can look for these kinds of giant effects, you know, through video visits, which will make it real easy for patients who have trouble traveling. And so how am I going to do this? Well, I'm partnering with patients like me, and they've got this amazing website. If you haven't been on there, I'd highly encourage you to check it out. You can go on there for free and create a profile that tells everybody about you and, you know, who you are, what your ALS looks like. But you can also put in there what things you're taking, and you can track different outcome measures, including the ALS FRSR. And so in these trials, we're going to teach people how to use this, get them registered, teach them how to measure their ALS FRSR, and have them go in there and do it. What's the advantage of that? Well, first of all, it's a lot cheaper to do it that way. But second of all, that's a publicly available database. People don't have to wait years to find out what's happening in one of these trials. They can go into patients like me at any time and see what's happening to anyone in one of these ROAR trials. And then the last part of all this is that I'm going to just publish the protocols in this program on the website, alsreversals.org. As soon as my IRB approves them, I'm going to publish them. And you might ask, you know, why would I do that? I would do it because I know there's people all over the world who are trying to self-experiment with things, and they're looking for guidance. And so I'll put this out there as a roadmap that they can take to their own doctor, and they can say, hey, could I try this like this person is doing in this trial? So they would be doing similar things to what we're doing outside the trials. So I've already done one of these ROAR trials. The first one I decided to take on was a study of a supplement called Lunasin. It's a peptide which is derived from soybeans. And the reason I chose this one first was because it looked like it did a lot of things in cells that were very plausible in terms of a mechanism. And there was a very dramatic and well-documented ALS reversal who was taking Lunasin. It also seemed to be very safe. So I did one of these ROAR trials, and there was some good news and some bad news that came out of it. First, the bad news. Lunasin was not nearly as safe as I thought it was going to be. It had a lot of GI side effects. It had GI side effects, including constipation so bad that I had to put people in the hospital to get them over it. There was no evidence that Lunasin slowed any of the 50 people that I had in this trial in terms of their progression. And there was no evidence that it reversed anyone else's ALS over the course of a year of treatment. The good news is that this unusual design worked very well. You know, most ALS trials enroll 1 to 2 patients per site per month, and this enrolled 9 patients per site per month. In most trials, there's a very narrow set of inclusion criteria, and so you tend to only study your drug in a small subset of people with ALS. And so even if you find something, we're never really quite sure, does it work in everybody else with ALS or only this one subset? In these ROAR trials, I'm getting a very diverse study population. There's some people who've only had the disease for a few months, and there's some people that have had the disease for 20 years. There's some people that have hardly any deficits, and there's some people that are quadriplegic on ventilators. And the reason I think that's so great is that I think you can generalize the effects of a trial like this across your whole clinic. So if I find something that works, I think it's going to work in everyone. You know, this design held on to patients all the way through the study better than most traditional trials. We showed that we could teach people how to measure their own ALS FRSR, and you know, with some training, they did it just as well as me and my coordinator. They would go onto the website every month like we asked them to. And by publishing the protocol, we had hundreds of people all over the world empowered to try something that we thought was reasonable for their ALS. And so we're gonna do more ROAR trials. And the one that we're just about to start is of another supplement called curcumin. So curcumin is a chemical, which you see there. It's an orange powder that has the chemical structure in the lower left-hand corner of the slide. There's at least 5 plausible mechanisms by which curcumin could help people with ALS. It's anti-inflammatory, it's antioxidant, it interferes with the aggregation of proteins, and maybe most exciting of all, at least in animals, it changes the microbiome. There have been preclinical studies in a cell model of ALS showing that forms of curcumin could do things to the cells that looked very promising. There have even been 2, not perfect clinical trials that were done in other countries in people with ALS that both showed benefits. 6 of the 48 ALS reversals that I found happened while people were taking some form of curcumin. And this appears to be very safe, and it's also very inexpensive, at least when it's taken orally. And so the design of this ROAR curcumin trial, I'm gonna get 50 people with ALS. I'm gonna ask them each to get me 1 healthy control from their same household. I'm going to treat the patients with curcumin for 6 months. I'm going to teach patients how to measure their ALS functional rating scale and their weight, how to teach them how to do perceived effectiveness and side effects. I'm going to have them enter that data every month on patients like me, and I'm going to compare the progression on these measurements to historical controls that we generate in the same way that we did in the Lunison trial. I'm also going to have both the patients and the healthy controls collect stool and saliva at 3 different time points throughout the study. And we'll be able to answer a lot of questions that we still have about the microbiome in the study. We'll be able to answer the question of, is it really different? This will be the largest study ever done. Is it really different, ALS versus healthy controls? Is it really different in people who have fast or slow progressing ALS, bulbar or limb ALS? Nobody knows the answer to these things. Can we change the microbiome with therakermin? And if we only change it in a subset, is that the subset that seems to respond to it? This will be a totally virtual clinical trial. There'll be no in-person visits. Everything's going to be done over video visits. The medication will be mailed. The kits for collecting saliva and stool will be mailed. Again, the results will be available in real time, and the protocol will be published as soon as it's fully IRB approved. It'll be a very easy study to get into. You can see the rules over on the right-hand side in terms of inclusion, exclusion. Tried to make it as easy as I can, but there's no rule here about how long you've had ALS or what your function is. So if you have a tracheostomy or attached to a ventilator, you're totally paralyzed, you're still a candidate for this study. So I'm gonna end now with this quote that I think applies equally well to ALS reversals as it does to me. And that is, there's no such thing as a weird human being. It's just that some of us require more understanding than others. ALS reversals, as I've shown you, they're rare, but I think they're important. I think they might be giving us clues to either some sort of endogenous mechanism that makes people resistant to the disease, or even some new treatment that's been discovered by these patients. The good news is I think I have a definition I'm comfortable with. I have several testable hypotheses. I've been able to find funding and collaborators around the world to help me study these ALS reversals carefully. I do want to also make this point. I think this is really important for folks to understand. We do not know why any of these 48 people got better. The treatments that they were taking are associated with their improvement. That does not mean they caused their improvement. There's no way to look at any one of these cases and say that this treatment caused this reversal. The only way to answer questions like that is through the clinical trials that I'm trying to do, those ROAR trials. So if you want to follow along, the Duke ALS Clinic has a Twitter feed that I try to keep up with. The ALS Reversals webpage has the protocols, and it will soon have the Therakerman protocol. We are IRB approved as of today. And as soon as we get posted on clinicaltrials.gov, we will be opening that study. I expect that study to open next week, and then we'll publish the protocol on our website if people want to try it. And I just want to thank a lot of people. I mean, first and foremost, all the patients and caregivers that are out there listening. I mean, you're the reason I get up out of bed every day, and I'm so excited to come into work. You're so inspirational, and you give me so many cool ideas to work on. And you know, you participate in the studies, you participate in funding the studies. I've got so many great families that have contributed to this ALS Reversals work. It's still very controversial. I'm probably not going to be able to get grants for it from groups like the NIH. And that's just because, you know, the reviewers are usually very conservative people, and that there's still a lot of controversy about this. But families like the one in this picture, the Larry Vance Hughes ALS Foundation, have stepped up and funded this work. And I couldn't do it without a lot of help from my colleagues. I mean, I've had two great medical students in the last few years, Dan Harrison and Jesse Crail, that did a lot of the legwork. And believe me, just validating one ALS reversal takes many hours. There's a lot of phone calls and emails and records being faxed back and forth. So thank goodness I have people like that to help me. And then finally, just a shout out to my Duke ALS care team. You know, they helped me build a great foundation of patient care at Duke. And it's really upon that strong foundation that all my research is built. So I'm happy to take your questions now. I'll turn off my screen sharing so that you can see me better.
Thank you, Dr. Bedlock. Thank you very much. That was fantastic. I mean, you know, you addressed a lot of questions that people usually have for us. So moving on to the next session, we do have Mick Finn, whom you know very well. You're number 42. And you know him not only as a person, you also know his record. So he will be asking you and moderating the questions from the patients, along with Sarah Diaz, who is probably have exchanged emails with her. And I will let Sarah and Mick Finn, you know, take it away from him.
Perfect. So good evening, Dr. Bedlock. I want to start off with, which protocol resulted in the most reversals?
It's a great question, McFinn. Unfortunately, there are no two ALS reversals that use the exact same protocol. You know, the single treatment that's associated with the most different ALS reversals, the single, you know, thing that I think might be relevant is curcumin, and that's associated with 6 of these 48 reversals.
Thank you.
Oh, wow.
Sarah?
That's really interesting. Yeah, so based off that question for the curcumin, I know your study will come out with the protocol, and
Yep.
Patients are asking in the chat, does that include the dosage and the brand in that protocol, or are they going to have to find that part of the information?
No, and again, I think that's why I feel like it's so important to publish these protocols. You know, especially when it comes to supplements. Gosh, I mean, there's dozens of different kinds of curcumin, not to mention dozens, maybe hundreds of different manufacturers. I picked the one which is called Therakerman from the company Integrative Therapeutics, because there's just so much research that they've done. That's the exact formulation from the exact company that changed the microbiome in the animal studies. You know, that's the exact formulation that has really good pharmacokinetic studies showing that it gets into the blood and probably into the brain. So these are all really important things. And, you know, it's tough. I mean, people often from other countries will email me and say, I can't get that. you know, what about this one that I can get here? And the problem is a lot of these things haven't been studied. And, you know, to make matters worse, when people ever do like study supplements from different manufacturers, it's pretty shocking what they find. There's wild variation in what's actually in those bottles. And that's part of, you know, the problem with things not being tightly regulated by groups like the FDA is the quality control is not good. So yes, I will have all that in the protocol that I publish hopefully in the next week or so.
Wonderful. Thank you.
Thank you so much. We know that the reversals were about 10% genetic and 90% sporadic. Of those reversals with familia ALS, what were the common mutations seen?
Yeah, I mean, that to me is one of the most interesting questions of all. And I don't have the genetic data yet to answer it, but that was actually the question that I think sold this project to the folks at the CREATE Consortium. The fact that there are these 4 patients with a family history, and it's just, you got to believe that those 4 people must have an ALS causing mutation. And so the question is, what else do they have? What counteracted that mutation? It's just too much of a coincidence to think that someone with a family history of ALS got something that looked just like ALS, but it wasn't. Like, that's just too much of a coincidence. So hopefully by the end of 2020, I'll be able to answer that question, but I think it's one of the real key questions here.
Thank you.
In your presentation, Dr. Bedlak, you talked about the collateral growth as seen in polio, and as the nerve reinnervation. Do you think that any supplements that besides maybe curcumin could be playing a role in that reinnervation? And also in terms of that, were there comparisons in the amount of reinnervation to the increase in improvement in ALS FRSR scores?
Yeah, so those are both great questions. So it turns out to be really hard to study which supplements might cause reinnervation in people. Like, you know, you can only measure reinnervation very grossly with an EMG, like I was talking about. It's not a very fine way to measure the degree of reinnervation. So, you know, as far as like theories and things that are seen in cell cultures, the things that promote sprouting and reinnervation are usually what are called trophic factors, TROPHIC, also known as growth factors. So for example, like if you look across all the things we reviewed with ALS Untangled, there was one called IPLEX, which was a form of insulin-like growth factor. And that would probably promote sprouting in neurons in a dish. And so you would think that that would probably be something that might promote reinnervation in a person. But as far as actually proving that, it's I don't know of any way to do it. And, you know, the second question is a great one too, but it gets back to what I said. There's no real good way to quantify the exact amount of reinnervation and correlate that to the ALS functional rating scale. I mean, what I can say is a lot of the people in the ALS reversals cohort, when they had those EMGs when they were well, it didn't really show any evidence that the muscle didn't have any innervation anymore. Right. So it didn't really show any more denervation. It only showed reinnervation. So the sprouting was so profuse that it completely, you know, got the whole muscle connected again. So that's what I can say there.
All right. Do you have any theories as to why there are more reversals in men than women? Is it related to the fact that men have more exposure to industrial environment toxins at work? We know that people in the military have a high incidence of ALS. Are any of these reversals military personnel?
Yeah, so off the top of my head, I don't remember exactly how many, but I know at least one was in the military. As far as the male-female question, I mean, those are all really interesting theories. I mean, another theory that I think we have to consider is, you know, ALS affects men at a rate of about 1.5 times women. And that's about the same distribution of reversals. There's about 1.5 times the number of male reversals as there are female reversals. So I think I said there were like 38 males out of 48. So 1.5, you know, out of 48 would be 36. So we're very close. So I'm not sure that there really is a big difference. I think it's just that, you know, ALS in general is more common in men than it is in women. And so you would expect that reversals would also be that way, be distributed that same way.
Thank you.
Absolutely. So the next question kind of comes off of supplements and turns more to the pharmaceuticals. A lot of patients, and my dad included, have tried the Radicava and the Riozols. And were there any reversals using other drugs? Maybe some of your off-label studies that you did that were approved by the FDA, but not necessarily just for ALS?
Yeah, I mean, it's a great question. So it's a hard one to answer because most of these ALS reversals were taking prescription medications. You know, I think I looked at it, it was like 30 out of 48 were taking at least one prescription medication, oftentimes for other problems like high blood pressure or cholesterol. But, you know, if you want to know about prescription medications that might be relevant, obviously relevant to ALS, yeah. So I believe there were 6 of them that were taking baclofen and a couple more that were taking Nuedexta. So yeah, I mean, there's a lot more work to be done to try to figure out if there's some combination of these things that in the right person, you know, might contribute to an ALS reversal, but it's not obvious looking at the list that there's an overwhelming number of one supplement or one prescription medication.
And limb onset?
Yeah, it's another great question. And, you know, no one knows for sure. There's a theory about sex hormones that, that they may play a role, but no one knows.
Okay, thank you.
More to find out in the future.
Yeah.
One of your studies in 2018 had to do with fish oils and omega-3s. So just for some of our patients, could you kind of explain why omega-3s would be important for PALS, as well as any differences between like fish oil or krill oil? Do they need to include a DHA or EPA? And is there any relation between omega-3s and ALS FRS scores?
Yeah, it's a lot of great questions there. The simple one is, no, I haven't seen any relation between omega-3s and ALS FRSR scores, but that's a question that we might be able to answer by looking at PROACT. So, you know, omega-3 fatty acids are pretty commonly used for people who want to try to protect their heart and optimize their lipid profile and things like that. So I suspect there'll be a fair number of people in PROACT who were taking omega-3s. The question is going to be like, how much more detail can we get? And that's actually something that I don't even have yet in my database, is how much more detail on, you know, exactly what was in the fish oil and whether it was truly fish oil or krill oil. So krill oil is very similar. It's also derived from tiny sea creatures, in this case, crustaceans, which are called Antarctic krill. And the difference is exactly what's in there. So krill oil actually has more antioxidants in it, and that's why it looks different than fish oil. It's typically a lighter shade of color. Like, for example, there's an antioxidant in krill oil, which is called astaxanthin. And so, you know, you can come up with theories why some formulations might be better. Like, sure, I mean, we always talk about oxidative stress in ALS, and, you know, we want to attack that with antioxidants. Maybe more antioxidants are better. The problem is that we've done so many studies of different antioxidants, and for the most part, they're not very impressive. And so I just wonder, like, I believe oxidative stress is happening in the brain and spinal cord, but I wonder, is it so late in the course of things, like so far downstream, that by the time you start to try to attack it, there's too many other pathways that have gone wrong. And so even if you found a really effective way to knock that pathway out, it may be that there's so many other pathways going haywire that the motor neurons are going to keep dying at the same rate. So, I mean, the short answer is, it's a very interesting question. I don't know the answer. I don't think there's enough evidence really to advise people to take any kind of fish oil for their ALS right now. I think it's definitely something that warrants further study, though.
That's interesting.
All right. Are there any specific protocols or supplements that are more effective for people with bulbar onset?
No, not that I've seen. I mean, there's nothing obvious that's emerging out of looking at, you know, these particular reversals. Now, one thing I can say, when you say effective, I'm assuming that you mean reverse ALS, but another definition of effective is, control symptoms. And so, I mean, getting back to Nuedexta, there's one more layer to that story that I told before. I talked about how we were studying it for disease progression. It didn't work. People in the study came forward and said it helped their laughing and crying. So it got approved for pseudobulbar affect. And since then, we've been using it for people with pseudobulbar affect. And we've noticed one more surprising thing. And that is that sometimes people that we give it to come back and their speech or their swallowing or their drooling or all three are much better. And it's not clear why this doesn't happen to everyone, but there was enough information where the person who has been behind this from the whole time, Richard Smith, who kind of invented this product, went back and did a small clinical trial. And he was able to show that objective measures of bulbar function actually improve on Nuedexta. So, when I talk to patients with bulbar dysfunction in my clinic now, I usually mention all this and I say, this is something that we can try. We have to officially prescribe it for pseudobulbar affect or insurance won't pay, but we can also hope that we might see some changes in some of these other parts of your bulbar function. And the improvements in my experience are usually seen pretty quickly within the first couple weeks. If I put somebody on Nuedexta in my clinic and I don't see their bulbar function improving, it doesn't seem like it's helping them in any way, I usually advise them to stop it. But that's just sort of an aside, that may be another level to that question that I didn't think of.
Thank you.
That would have been huge. My dad had bulbar ALS, so and we definitely struggled a lot with those kinds of symptoms. So I think now we get a lot of questions to our group about saliva issues. So that'll be interesting for people to maybe try and take back. One of the reversals that you have studied used a holistic NRF2 activator. And so I was kind of hoping that you could talk to patients exactly what that means, just in case they review his information, as well as the new ALS study about Tecfidera. Do you know of any of your patients that are taking it? And could they discuss this with their neurologist as an option?
Yeah. So NRF2 is actually a molecular pathway that's kind of like the equivalent of flipping a switch in cells that makes cells start to produce more antioxidants and more anti-inflammatory substances. So it's a really cool pathway to think about manipulating in a disease like ALS. So, I definitely know that there are prescription drugs, like for example, Tecfidera is a prescription drug that is approved, FDA approved, for use in multiple sclerosis. And it works by activating this pathway. And what it does for people with multiple sclerosis is it decreases the number and frequency of attacks that they have, where their immune system attacks parts of their brain and spinal cord. And over time, they wind up accumulating less disability. Now, of course, the situation in the brain and spinal cord of people with ALS is not like that of MS. It's not as clear that inflammation and oxidative stress are definitely driving the death of motor neurons. These are theories. But there is that one ALS reversal who took a product, Protandim, which is an NRF2 activator, and shortly after starting that, began to recover the ability to take care of himself. Had lost the ability to dress and feed himself, and after a few months on Protandim, recovered that. And that's very, very rare in my experience for people to have those kinds of changes. And it's enough to call it an ALS reversal. Now, people might ask, well, could I get Tecfidera since it's approved? Yeah. Any doctor can write a prescription for Tecfidera for a person with ALS, but there's some things to remember. So first of all, Tecfidera, it's been carefully studied in MS, and it is known to have many potentially serious side effects. It can drop the blood counts. It can damage the liver. It can also cause a life-threatening brain infection, which is called progressive multifocal leukoencephalopathy, or PML. The other thing about Tecfidera is very expensive. I doubt very much that insurance would let that slide through, without an MS diagnosis. If an insurance company saw that a doctor wrote a prescription for that, they would say, okay, what's the diagnosis code? And they would see ALS and say no. And so, you know, if you had to pay for that out of pocket, I think it would be around $3,000 a month to take it at a similar dose to an MS patient.
Oh. Has research been done on cannabis for ALS, for example, for saliva secretion reduction or stress?
Yeah, so the short answer is there's a lot of anecdotal data that suggests that cannabis products could be very helpful to people with ALS and PLS. So I have seen hundreds of patients over the past 20 years that were on some form of cannabis. And I'm convinced that many of these people had improvements in their appetite, their muscle twitching, their muscle cramps, their anxiety, their sleep. These are all symptoms that are challenging for patients with ALS. And, you know, we don't always have the greatest things to offer. I mean, we have things we can offer, but sometimes those things that we use have side effects. You know, for example, severe pain is sometimes managed with narcotics, and we know how many side effects narcotics can have. Wouldn't it be nice to have another option like cannabis that seems to be so much safer than some of the things that we use? You know, we're only going to get it if we get some clinical trials in people with ALS, not just anecdotal data, not just trials from people in other diseases. And, you know, it's very difficult to do clinical trials of any cannabis product in the United States because of the laws. Surprisingly, you know, even in states where cannabis is very legal, very easy to get, the Drug Enforcement Agency nationally still considers it illegal. And so I've talked to some of my colleagues who practice in those states and said, hey, now that it's so easy to get in your state, would you do a clinical trial? And they say, we can't, because, you know, we have to use our DEA number, you know, to prescribe things for patients. And if the DEA finds out that we're giving out cannabis, they consider it illegal. They could take away our DEA number, we might lose our license. So the whole thing is nuts, in my opinion. The good news is in other countries, they're starting to finally do ALS trials. I know of one in Australia. I know of one in Canada. And my plan is, you know, if those trials show evidence, whether it's symptom benefit, whether it's slowing of disease progression, I'm hoping to get, you know, a petition and a big advocacy effort to try to get this legalized for people with ALS. The good news is we wouldn't be the first ones to do this. There's actually another disease that happens to kids. It's called Dravet syndrome. It's a really bad seizure disorder that some kids get. And, you know, people associated with that got data. And across the whole United States, you can prescribe very potent cannabis products to kids with Dravet syndrome legally. So there's a precedent for this. If we could get the data, I think we could actually, you know, get this exception made for people with ALS, but we're going to have to wait for the data.
Okay, well, I use cannabis every day during my ALS, and the doctors gave me painkillers and antidepressants and sleeping pills. And I called up the doctor the next day. I said, you know, I took those last night and I didn't feel good about that. Can I just go to cannabis oil and marijuana? And the doctor said, if you feel that sincere about that, you go right ahead. And
Yeah.
So I just want to say that. And one more interesting thing, I was a toy maker for 21 years.
Oh.
So I had a wood shop. I wanted to be Santa Claus when I grew up. And so I started a toy shop. So that woodworking, I breathed so much sawdust that finally one day I said, you know, I'm going to stop now. I think I'm going to sing to people instead of giving them wooden toys. So anyway, just wanted to throw that in there.
Yeah, you know, McFinn, your first comment, you know, just brings up one of the reasons why it's just so important that we start to study this, right? Because, I mean, I'm already convinced that many of my patients have had symptomatic benefits, but I sure would like to know more about, you know, what is the best strain of cannabis? What is the best route? What is the best dosing? And I would like to also know, does it actually have any effects on disease progression? And I think we can only learn the answers to those things through trials. Otherwise, it's pretty random.
Okay,
Well, I think you found your patient advocacy group to do it. I mean, McFinn will run that 100% and he'll use everything ALS to do it too.
I
You
would
got
love
it.
Awesome.
Okay.
So since that would be through ALS Untangled, most likely, we'll talk about some more ALS Untangled studies. One of your 2018 studies was with glutathione.
Yeah.
Were there any particular methods of delivery seen in the reversals, and is there any one that you recommend specifically for neurons, as well as is there a difference between that IV glutathione NRF2 applicators?
Yeah, I mean, it's a fantastic question. So glutathione is the main antioxidant that's in all of our cells. And, you know, you may be able to increase the amount of glutathione in cells by giving intravenous glutathione, but it's pretty clear you can't do it by giving oral glutathione. Now, there may be other ways, other oral products that can increase the amount of glutathione in cells. There's glutathione precursors, for example, that people might be able to take. But here's the problem with all this. There already were clinical trials of IV glutathione as well as some of these precursors in people with ALS, and they didn't show any benefit. So if a very potent way of increasing the amount of glutathione in cells doesn't produce benefit in a trial, it's just not something I'm gonna be that excited about offering to my patients. I mean, I feel like, we start with things that, maybe have plausible mechanisms and some anecdotal data and look safe. But if we already know there's a trial and it didn't work, I just scratch it off the list. I say, well, let's move on to something else. There's so many other things out there that we could try that haven't been tested in a trial and shown not to work.
Absolutely.
All right.
Thank you.
Gunther, was your 44 reversal? Have you had a chance to review any parts of his protocol that he used? If so, can you tell us what parts may have valid clinical mechanism?
So I'm going to refer to ALS reversal number 44, since I don't have permission to share every participant's names. But ALS reversal number 44 was experimenting with a peptide therapy that he was getting from a clinic in Florida, a myostatin inhibitor, nicotinamide dinucleotide, NAD, as well as a spiritualist. And so of all these things, NAD definitely, has the most plausible mechanisms. And we wrote about those in our ALS Untangled review of a product called Basis. The other things in this regimen, I mean, I think the mechanisms are very thin. I mean, they're probably not going to get very good grades when we actually get into reviewing some of these things. So it's not clear to me, exactly how a peptide injected, or taken orally could help a person with ALS. I mean, peptides are combinations of amino acids, and several different amino acids have already been studied in ALS and didn't seem to do anything. And, myostatin inhibitor, well, myostatin is, it's something that is involved in muscle. And so if you had a muscle disease, I could see that might work. But when the problem is, dying motor neurons, I'm not sure why that one would work. So of all the things in that regimen, to me, the NAD is the most mechanistically plausible.
Thank you.
Well, that totally brings up the next question. Vitamin C and NAD are not officially reviewed, just as like you said, but can you kind of explain for people that might be newer to this journey what NAD is, and a new precursor that's called EH301, kind of why that's gaining some interest and what might be beneficial to ALS patients?
Yeah, we'll start off with the easier of the two, and that's vitamin C. So, vitamin C is yet another antioxidant, and a lot of people take it to try to ward off things like colds. But the idea of using this in ALS is not new. There was a trial done many years ago, actually a case series, and unfortunately it didn't really show any benefit. That people in that paper progressed at the same speed on vitamin C that they were progressing at before they started it. So again, that's one that, for my own personal list is not near the top. And then there's this product EH301, which is related to a supplement called Basis. And when I say related, well, the two things have the same ingredients in them, The question is, are they present in the same amounts? And that is a question which I still do not have the answer to. So the company that owns both of these products, which is called Elysium, has been very quiet about publicly releasing exactly what the difference between EH301, which was in a clinical trial, and BASIS might be. So EH301 was studied in a clinical trial. It's a very strange trial, but at least it was a trial. At least it was published in a medical journal. It's strange for several reasons. First of all, it's very small sample size. You had 15 people that were assigned to get this EH301 and 17 assigned to get a placebo. And it was randomized and it was blinded. Unfortunately, as sometimes happens in a randomized trial, when you look at how the groups were doing before they started treatment, they're not exactly the same. They're not well balanced. So it's not clear that these groups would have done the same no matter how they were treated. And that, when you have a really small trial, sometimes randomization unfortunately winds up that way. It's not that anything was done on purpose. It's just an effect of randomizing a small number of patients. The most difficult thing I have with this trial is the duration. I don't know that I've ever seen another ALS trial that was this short. It was only 4 months. And most ALS trials are at least 6 and more often 12 months. And you had a lot of dropouts. 5 out of 15 people in the drug-treated group dropped out in the first 4 months, and 7 out of 17 in the placebo group. And we were told that the dropouts were not because of adverse events, but there really was no explanation given. One of the dropouts was said to have hepatitis. One of the dropouts was said to have profound mental depression. And it's not clear, why did they get those things? Did they have those things before the study? Did those things start during the study? How do you know they weren't related to the product? And then finally, when you have dropouts, you're supposed to account for them when you analyze the data through something called intention to treat analysis. You're not supposed to just analyze the patients who finished and did well. That's considered too optimistic of an analysis, and that wasn't done here. But the bottom line is that the trial did show a benefit. So it showed that on many different outcome measures, the small group of people that were on this EH301 seemed to be stable or even better than they were at the start of the study, and everybody on placebo was worse. And so even though there's many flaws in the study, it's very interesting result. It's very exciting. And I hope that somebody's out there trying to replicate this in a much better design. Because anytime we get a positive trial, we gotta jump on it and try to really figure out if it's real.
Absolutely.
You mentioned the microbiome. Do you recommend pals take pre and probiotics? What's the best way for them to test to see which good bacteria they may be missing in their gut?
Yeah, Finn, it's a great question. And I hope to have the answer to that question, but I think it's going to take me a little time to figure it out. And, so as I mentioned, there have been these 3 studies, very small studies, that all showed that the microbiome was different in people with ALS compared to healthy controls, but the differences were not consistent or clear. And I think it may just be the small numbers. Everyone's microbiome is different. So I'm in the Theracurmin study, hoping to really help answer the question. A hypothesis that I have is that there is this one bacteria called Akkermansia eosinophilia, which produces butyrate, which is neuroprotective. And that's the one that they inserted into the animal models of ALS that slowed progression. And there are some other bacteria, Ruminococcus and Puribacteroides, that produce things that would be expected to maybe ramp up neuroinflammation. And when they injected those into the animal, they put those into the microbiome, it dramatically sped up progression. And so my theory is that maybe people with ALS who are doing really well have more Akkermansia, and people who are progressing quickly have more of these other kinds of bacteria. And hopefully something like Theracurmin can shift the balance. But I think only when we got more data can we start to think about, what would a rational probiotic regimen look like? What would it include? What are we trying to put in? What are we trying to get rid of? We just need more data.
Absolutely.
Thank you.
On ALS Untangled, sodium chlorite was studied, and we know that a couple of the reversals used it. Can you just kind of talk about generals behind sodium chlorite and if there's been any follow-up with those reversals on how they're doing and if this could be something to look into again in the future?
Yeah, so sodium chloride is a molecule that can block the activation of macrophages and microglia, which are part of neuroinflammation. And like we were talking about with curcumin, it isn't just one thing. So there's different products. There's different intravenous forms, and then there's oral forms. And so when we wrote the review, we talked about a patient with ALS who had tried an oral form and had reported that they had some benefits. Now, unfortunately, that was one of those transient improvements. So that person wasn't included in the ALS reversals. They're not one of the 48. So none of the 48 ALS reversals in my program were on any form of this. But this is another really good example of why we have to be very careful. Like when this study first came out and it suggested that maybe there was something going on here positive, that some people might benefit from this, immediately a lot of people online started talking about taking oral sodium chloride. Well, it turns out that the study used IV, a very specific IV form. If you look at what happens with oral sodium chloride, that molecule gets changed immediately by the saliva in the mouth. And it actually gets changed into something that's potentially poisonous. And that's why the people who were developing this drug in the first place immediately switched from developing an oral formulation to an IV. Because I talked to the guy who invented this, and he said the first couple of people that they gave the oral formulation to collapsed, became unconscious. So they said, that's enough of that. And they later figured out why. But it's just so important. Drug development is hard. And just because something has the same name doesn't mean it's the same thing. But the last part of the question is, what happened to this? And as you probably know, the company went on and did a much larger trial, which for some reason did not show the same benefit. And all I can think of is that, once again, it's the danger of very small numbers. When you're looking at very small numbers, and we talked about how the disease is not a linear progression, all you would need is a couple of those patients, who had those long plateaus to wind up in one group or the other in a small trial. It could totally change the results of the study. When you do a much larger trial, you kind of wash all that out. But the larger study didn't show a benefit, and I don't know of any more plans, to do anything more with that molecule.
Right. In a recent hearing, Congressman Fortenberry discussed your support for expanded access in clinical trials. For people on this call, can you outline your reasons for supporting it and why you don't have concerns about the safety for PALS?
Yeah, so as many of you know, it's an amazing time as far as advocacy goes. There's been so many incredible things that patients with ALS and families have done over the last few years to change laws. And there's a bill before Congress now, which would create a pot of federal money for expanded access programs for people with rare diseases, terrible diseases like ALS. So I just felt like I needed to write a letter to the congressman to express my support for this bill and to help him to understand, my perspective on it. And so my perspective on it, again, comes from 20 years, about 3,000 patients seen. Almost every person I've ever seen with this disease wants to try things. They want to fight the disease. Of course. If I got the disease, I would want to fight it. I would want to try things. And, what things do they want to try? Well, many times they want to try the things that are in trials, because those are generally thought to be the most exciting things. They have the most preclinical evidence. There might even be some early phase, human data suggesting that they're of benefit. And so you might ask, well, why don't they just join the trials? So the problem is multifaceted. First of all, there's not nearly enough spots in these trials. If we put all the ALS clinical trials together, there may be, 2,000 spots, and there's 20,000 people with ALS in the United States. So the vast majority of them are not going to get a spot. Second, many times patients don't qualify for trials. Like some of the trials that sponsors bring to my clinic, only about 5% of all the patients in my clinic would look like what they want for the trial. Third, many times patients don't want to put up with all the things that the trial's asking them to do. Many trials have lots of visits, many trials have placebos, and many times patients say, I just don't want to put up with all that. I just want to take something and see if it helps me. And so the only way that we're going to get through to something like that is by expanded access. And so in order to get one of these products that's in development, 4 things have to happen. The patient has to ask for it. Their doctor has to be willing to give it to them and supervise the treatment. The company that owns the product has to be willing to give it to the patient. And finally, the FDA has to approve the whole thing. And so you may remember in the last couple of years, there was this tremendous movement to create a right to try law. And, it's a testament to all the hard work that not only passed in almost every state, but it passed federally. So there's a federal right to try law. And what happened when that passed? Did all the patients who wanted to try things get to try things? Unfortunately, almost nobody did. And the reason is that law, it attacked the wrong part of this 4-step problem. It attacked the FDA. The right to try law basically said the FDA should have nothing to do with this. Well, the FDA was never the problem in the first place. That was not the reason why so many people cannot get access to these products. Ever since the early days of HIV, if you watch the movie Dallas Buyers Club, you can see that the FDA stepped up and created something called expanded access programs, where people could try things as long as there was some sort of protocol and some sort of plan for monitoring and supervision to protect the safety of patients. The FDA's had these programs, for more than 20 years, and they've approved almost 100% of the applications that have been made for expanded access. And they usually approve it very quickly. They can approve it within 24 hours if the doctor asks them to. So the right to try law attacked the wrong part of all this. Patients want it, the FDA will allow it. It's those 2 other steps where we got problems. And let me explain why. So the problem from a clinician standpoint is that it's a tremendous amount of work to get these things for patients. It's not that we don't want to do it. I currently follow 400 patients. What if I go in tomorrow and I have 250 messages that 250 patients want to try 250 different products? Well, that means 250 different forms that I have to fill out for the FDA and for the IRB, 250 different shipments of products that I've got to figure out how to receive and distribute to patients. And there's no funding for that. That would all be done like between the hours of 8 p.m. and 6 a.m. when normally I try to eat and get some rest. So, you're stretching, really stretched clinicians. And then there's the companies. And the companies, what is their incentive to give these products to people? Some of these small companies have invested everything they have in the development of these products. It's very nerve-wracking to think about giving these outside of the trial. Companies may fear that something bad is going to happen that could destroy, their ability to get the drug approved, even though that's extremely unlikely. It's actually never happened. Companies may not actually have enough of the product. They may only have enough of the product to do their trial. And so what I love about this law that's in front of Congress is it finally actually targets those middle 2 steps, which are the reason that people can't get expanded access. This pot of money could be used for physicians to maybe hire some infrastructure like a coordinator to help them with all this, and for companies to actually be able to get some money to make more of the drug. So I think that this law has a really good chance of actually finally fixing this problem and allowing more patients to get access to things that are in development. That's why I wanted to write a letter and support it.
Well, for anybody who saw our talk with Congressman Fortenberry and Congressman Quigley, we definitely support Dr. Bedleck in writing letters of your own. So we will send out that information again so that we can also show our support as patients and families in order to help these middle two steps, like Dr. Bedleck was just talking about. This is kind of on the other side of the spectrum, but your study about clenbuterol, can you just kind of talk about why you think this drug is going to be beneficial, maybe even to bulbar patients?
Yeah, so I mean, it's got a really interesting mechanism of action. So this is actually a drug that does stimulate a growth factor. It stimulates insulin-like growth factor, and you can measure that in muscle. And it's been shown in preclinical studies that it can make muscles get bigger and stronger. And so outside the United States, it's approved for the use in asthma. In the United States, it's not approved for anything, but if you're a sports fan, you may have heard of it because sometimes athletes use this to try to cheat. And if they test positive for clenbuterol, it's considered a performance enhancer. They get a ban for some time period from their sport. And there's been several high-profile examples of being banned for what they call clen, C-L-E-N is what they sort of the slang word for it. So, I mean, that's an interesting mechanism, but what really got my attention was that there was a previous very small, again, flawed trial that was done in Italy more than a decade ago now. And it showed that a small group of people with ALS who took this for 6 months actually had improvements in several different things, including their breathing, their forced vital capacity went up over the course of 6 months, and also their muscle strength. So when they measured their muscle strength in their arms and legs, it improved. And I just don't, remember seeing very many trials in my career where people were followed for 6 months and on average had improvements, not just slowed progression, but actually were better at the end of 6 months than they were at the start of the study. So we've got 19 people enrolled in the clenbuterol trial now. Again, we kind of got stuck a little bit with new enrollment during the pandemic, but we're back, we're enrolling. We've got everybody scheduled. We're going to be studying 25 people for 6 months. And so we should very quickly have the last 6 enrolled. And, roughly 7 months from now, we should be able to release some information to the community and let everyone know whether it looks like this really does work, whether we were able to replicate the results of that Italian trial.
That's wonderful.
Thank you. Let's get real basic here now, Rick. Is there any correlation between daily lifestyle and ALS reversals? Does exercise play a role in these lifestyle protocols?
Yeah, I mean, McFinn, that's a great question. And again, that's one where we'll have to think carefully how to study that, because for people whose reversal just happened in the past year, they probably have a pretty good recollection of everything they were doing, their diet, the amount of exercise, the amount of stress they were dealing with. But when you ask someone to think back 30 years, what were you taking over those 2 years as far as your diet and what was going on in your life? It's probably pretty hard to remember all that. But it's not obvious to me right now, but it's not something that we've carefully studied. And the reason is I don't quite know how to study it yet. And I'm certainly open to ideas if people have, studies that were done in other fields that they know about where they tried to carefully look at all this. I'd be happy to take a look at those and see if we can apply that to the ALS reversals. I'd love to study it. I just don't know how to do it yet.
Do you see anybody exercising? Do you promote that?
So I do promote it in all my patients. It's pretty clear now. I mean, again, one of the many things I was taught when I was a resident, my teachers were saying exercise is bad, but now there's been many studies, and the studies clearly show that people with ALS who exercise feel better and probably progress more slowly compared to people who don't exercise. The questions that we don't have answers to are, what is the best kind of exercise? How much exercise should someone do? Is it possible to do too much exercise? So what I usually tell my patients is, look, I want you to try to exercise every day. We need to find something that you enjoy, something that you can do safely, and we need to start out very gradually and titrate it up. And if we get to a point where the next day you're in pain or you're exhausted, that's probably too much. We need to titrate back down to the next level. But I try to get all my patients to exercise every day. And yeah, I mean, having talked to most of these ALS reversals myself, I can say that in general, most of them were pretty active and were doing some sort of exercise, but I haven't carefully studied that and compared it to patients, you know, with more typical ALS and other databases, because I don't know how to study it. I don't know what kind of instrument to use yet.
Okay, well, thank you. Because 14 years ago, they told me don't exercise. It's not going to do you any good. And I was sad about that. So I'm glad things have changed.
Yep.
Thank you. Sarah?
So Dr. Bedlack, we have about one more question, but as a follow-up to your clenbuterol, we have a couple patients on the chat asking, is it a potential for them to be able to be enrolled in the clenbuterol trial?
You know, you can certainly email me and we can put you, we do have a waiting list. Right now, we only had funding to study this in 25 people for 6 months. And so right now we do have, you know, the 19 are in and the other 6 spots are scheduled. And I know there are some people on a waiting list in case one of those 6, you know, changes their mind or doesn't qualify. But I'm always happy to put people on the waiting list. I mean, I've got no problem putting people on a waiting list for any studies.
Oh, wonderful. And our final question comes directly from the chat. A couple of our patients were wondering, if a patient has a diagnosis but never progresses to full disability, are they not considered a reversal, more of a stabilization, or how do you classify reversal versus stabilized?
Yeah, so I think that there's something called plateaus where people with ALS stop progressing. And as I showed you in that PROACT data, most of the time, you know, that happens just over a short period. But there are definitely people who have plateaus that can last for years. I mean, I didn't ever get to meet Stephen Hawking, but I've read a lot about his life and, you know, watched everything I could get my hands on, all the movies about his life. And it really seems to me like he progressed a lot for like the first 5, maybe 10 years. And then for like the next 30 to 40 years, it doesn't seem like he changed very much at all. It seems like he had a plateau. And I personally think there's probably a lot more plateaus where people just stop progressing than there are dramatic reversals where people recover a lot of lost function. And I would love to study the plateaus. But right now I'm pouring everything I have into these reversals. If I learn some things from the reversals, hopefully, it will teach me what to look for in the plateaus as well.
Absolutely. Well, we want to first off, thank you so much for your time. This has been just overwhelmingly exciting information, and I think really has helped answer a lot of questions that we have received as well as been seeing online that patients want to know in terms of next steps for their progress in their journey. We're going to go to our open forum where everybody can communicate and talk. And Dr. Bedlack, we encourage you, you're more than welcome to stay, but we want to respect your time. So if you need to go, we totally understand. But again, thank you so much. And I do want to say in August, we're starting our care month. So we have been so filled with cure information these last few presentations from Dr. Merritt and Dr. Rothstein and Dr. Bedlack tonight. And starting in August, we're going to switch to the other end of our mission and talk about care and speech therapy and things like that. So we encourage everybody to continue to come. It'll be in 2 weeks on Wednesday, and learn some information on how to care for themselves in their day-to-day. So thank you again, and we hope to continue to see all this great work and make that 48 keep going up and up and up.
Well, thank you so much for having me, Sarah. I'm probably going to sign off just because my voice is about to go. But please feel free to email me any other questions that come up. And, if I can do it again for you in the future, hopefully in a year or so, I'll have a lot more answers about these reversals.
Thank you so much, Dr. Bedlack. And, we had a record, probably about 400 people today joined to hear you. So we're really excited. And also we want to talk to you offline about, how do we help with this funding bottleneck? If you believe in something so much, we want to be able to, work with you on some of those aspects. So with that, I would say good night to you. And thank you so much for sharing your evening. And we are honored to have you here.
It was my pleasure, Indu. Thanks for having me. Take care, everybody.
Thank you, Doc.